Hello, everyone. Welcome to Axiom Healthcare Strategies' latest town hall on heme malignancies. My name is Wajiha Ali. I'm a director at Axiom. I look at the heme space. I'm joined today by our esteemed panel of leading experts in the heme malignancies. We'll allow everyone to join in and give a minute before we introduce our panel formally. But a little background about Axiom Healthcare Strategies. We are a think tank style consultancy based out of Princeton, New Jersey. We look at the oncology and rare disease space, and some of our solutions help leading pharma companies. We work with medical centers and cancer research institutes. And our footprint is fairly global with our network of opinion leaders across the globe. Our therapy area coverage includes solid tumors, heme malignancies, as well as rare diseases. So, and I'm joined also today by my by our managing director, Mr. Hafiz Sikdar, and we look forward to a good discussion today. All right. So our panel today is Dr. Etienne Stein from Memorial Sloan Kettering, who will be discussing AML, myelodysplastic and myeloproliferative disorders, as well as CML. Dr. Nupur Raje from Massachusetts General Hospital, who will be covering myeloma and plasma cell disorders. And finally, Dr. Matthew Madisar from MSKCC again, and he will lend his expertise on lymphoma. Please join me as I extend a warm welcome to our panel. Can we move to the next slide? Yeah, the one before. Our agenda today covers 4 broad sections. First, our panel will discuss the latest updates across different malignancies. These will mainly be data that was presented at ASH annual meeting past December. We will cover both the practice impacting data that shaped the landscape of treatment, as well as some of the advances in the discovery of novel targets or development of innovative drug constructs such as cell therapy. Next, we will move on to discussing the outlook for these indications. And then our third section will be essentially the impact of the ongoing COVID-19 pandemic as it also continues into 2021. Lastly, our experts will take your questions. There is a Q&A and a chat functionality in this in Zoom. Please feel free to keep adding your questions. We will start answering them either between the presentation or towards the end. Thank you. Over to you, Dr. Stein.
Thanks so much, Wajiha, and thank you to Axiom for inviting me to be part of this panel. So there's a lot that's going on in the field of myeloid malignancies, much more than was going on even 6 to 7 years ago. So this is really going to be a whirlwind tour in 10 minutes of sort of the major highlights, both of things that were practice changing in 2020 and also things that occurred at the American Society of Hematology annual meeting. So the first thing is looking at Vyxeos. Vyxeos is a liposomal formulation of 7 and 3. It was shown in a randomized open label phase 3 study to be better than 7+3 in terms of overall survival in patients who had therapy-related AML and AML arising from a prior myelodysplastic syndrome. At ASH this year, the 5-year results of this trial were presented. And the important part of this, I think, is in the upper right-hand corner. You can see that the curves showing a survival benefit continue to separate. They don't come together at 5 years. And perhaps more importantly, when you look at the bottom right-hand corner, what you can see is that the overall survival of patients who were landmarked from the time of their allogeneic stem cell transplant, that is those patients who achieved a CR or a CRI, then went on to an allogeneic stem cell transplant, benefited those patients who received Vyxeos. Next slide. Oral azacitidine, otherwise known as CC46, is exciting because this is the first drug that has been shown to lead to a survival benefit in a very well-performed, randomized, placebo-controlled, double-blind study for maintenance therapy in patients with acute myeloid leukemia. So we never actually had maintenance therapy for AML. This study shows an overall survival benefit for patients with AML who did not have an allogeneic bone marrow transplant, who received CC486 compared to receiving placebo, the median overall survival of 24.7 months in the CC486 arm, and the median overall survival of 14.8 months in the group of patients who did not receive CC486 and received placebo. Next slide. Now, the IDH2 inhibitor, IDIFA, otherwise known as enasidenib, was approved based on the results of a single-arm phase 1/2 study, and the approval was based on duration of response, rates of complete remission and complete remission with partial hematologic recovery, and the rates of transfusion independence. So in relapsed refractory patients with an IDH2 mutation, this had become or has become the standard of care. There was a randomized phase 3 study that was presented in the form of a press release showing that in this randomized study, open label, when patients greater than 60 with relapsed and refractory AML were randomized to receive enasidenib or conventional care regimens, that is investigators' choice of chemotherapy, the median overall survival was not different between the two arms. We don't know really a huge amount about this trial yet, other than a 2-line press release. It'll be very interesting to see what the details of this are and whether there were any subsets of patients who actually did benefit from enasidenib. Next slide. Magrolimab is an anti-CD47 monoclonal antibody that has gotten a lot of play and for which there's a lot of excitement in a variety of malignancies. This is a trial giving magrolimab with azacitidine in patients with high-risk myelodysplastic syndrome and acute myeloid leukemia. And the big deal here is that the overall response rate in these patients with newly diagnosed acute myeloid leukemia was 63%. And maybe more impressively, the group of patients who had P53 mutations, that's a group of patients who are very, very difficult to treat and to cure, they had an overall response rate of 69%. You can see the survival curves both on the left and on the right. I'm not sure whether you should make too much of those because the median follow-up isn't very long, but the response rates are quite impressive. And this is a drug that is now being explored, particularly in patients with P53 mutations. Next slide. Now, menin inhibitors are small molecule inhibitors of the menin protein. And the menin protein has been found to be very important in patients in the pathogenesis of acute myeloid leukemia in patients with MLL rearrangements and in patients with NPM1 mutations. There's actually really good preclinical data showing the efficacy of menin inhibition in these patient subsets. This is clinical data from one of the companies that has a drug in clinical trials. This is KO539 from Kura Oncology. They only presented a few patients, but I think the thing that's pretty exciting here is number one, that in that 200 milligram cohort, you can see that they have a patient with an NPM1 mutation that achieved an MRD negative complete remission. And maybe more exciting, in the 100 milligram cohort, there's a patient who achieved a complete remission that was MRD positive. And this patient had neither an MLL rearrangement or an NPM1 mutation, at least raising the possibility that menin inhibitors could be used outside of where the preclinical data has shown them most effective. There's another compound that's being developed by Syndax that's also in clinical trials. They had some clinical data presented at AACR last year. And I think menin inhibitors as a general class are something that's going to really come to the forefront in the next 1 to 2 years for at least the MLL rearranged NPM1 mutant patients and perhaps beyond that as well. Next slide. Pevonedistat. So pevonedistat is a NED8 activating enzyme inhibitor. And there has been an ongoing clinical study looking at the use of pevonedistat with azacitidine in patients with higher risk myelodysplastic syndromes. So this is a clinical study that randomized patients with high risk MDS to either pevonedistat and azacitidine or azacitidine monotherapy. Unfortunately, this clinical study, at least in the primary analysis, was negative, with the median overall survival being statistically equivalent between the PEV-AZA group and the AZA group, although event-free survival favored the PEV-AZA group. It's going to be interesting to see what happens with this compound. Another thing that's important to point out, and is important for myeloid malignancies in general, and maybe all of cancer, is that the remission rates are much higher in the PEV-AZA group. But having a higher remission rate in a group of patients with myeloid malignancies often does not translate into improvement in overall survival. Next slide. A drug which I can't pronounce, but I'll just go by its former name, which was APR 246. So this was a compound that was said to be a reactivator of mutant P53. It's a drug that's been around for a long time, and there's a preclinical hypothesis about how it reactivates mutant P53. And based on this preclinical hypothesis, Apria Therapeutics performed a randomized study comparing APR 246 with azacitidine versus azacitidine monotherapy in patients with P53 mutated myelodysplastic syndromes. The primary outcome of this study was the rate of complete remission. And again, what we see here is that although there was very exciting phase 1/2 data in this phase 3 study, the combination of APR 246 and AZA plus AZA versus azacitidine monotherapy, although it shows an improvement in the CR rate, that improvement is not statistically significant with a p-value of 0.13. And similarly, the duration of response was very similar, at least statistically equivalent between both arms. It's a little bit hard to know where APR 246 is going to be going based on these results. They do have some other trials that are ongoing. Next slide. Cusatuzumab. So cusatuzumab is an anti-CD70 monoclonal antibody that is said to target the leukemia stem cell in a very small phase 1 study that actually got published in Nature Medicine with 12 patients. The overall response rate, and I think it was actually the complete remission rate also, was about 90%. But when you do a larger trial, those complete remission rates often go down. And this is actually a press release. This actually wasn't even presented as a paper. It was a press release that was slipped into a annual, actually a financial update report a few weeks ago. And you can see that in a larger study, the phase 2 trial of cusatuzumab with azacitidine, the complete remission rate here has dropped to 27%. And if you sort of broaden your definition of complete remission and you include those patients with incomplete hematologic recovery, the number goes up to 40%. There's an ongoing study of the triplet of cusatuzumab, azacitidine, and venetoclax. And what the company has said is that they're going to wait to see the results of this study before they make any further decisions about how to move this compound forward. Next slide. STAMP inhibitors. So STAMP inhibitors, specifically this drug, asciminib, is a drug that is used for the treatment of Philadelphia positive disease, so CML. And this was a study that randomized patients to receive either asciminib, which was actually known as ABL001, or bosutinib in those patients who had previously been treated with greater than or equal to 2 other tyrosine kinase inhibitors and either failed or were intolerant of those tyrosine kinase inhibitors. And the overall takeaway here is that asciminib is significantly better than sort of your bosutinib, whether you look at cytogenetic complete remission rate, you look at major molecular response at 24 weeks, or what's known as an MR4 and MR4.5, that's the log reduction in the rates of the BCR-ABL transcript protein. Next slide. And I think finally, we're going to talk about bromodomain inhibitors. So bromodomain inhibitors have been around for a while and have been investigated in a variety of myeloid malignancies. They may have found their home, though, in patients with myelofibrosis. So we're going to see a couple of clinical studies here. This is a clinical study of CPI-0610, where they combine CPI-0610 with ruxolitinib, with the JAK2 inhibitor ruxolitinib, and patients who are JAK inhibitor naive. And the outcomes they're looking at is the spleen volume response rate, and they're looking at the total symptom score. And you can see on the right-hand side in the waterfall plots that when you give these two compounds together, you get very nice spleen response rates. You also get very nice changes in symptoms. But maybe more impressively, if you move on to the next slide. So this is a clinical study in patients who were on ruxolitinib, but these patients did not achieve an adequate response on ruxolitinib. So the bromodomain inhibitor CPI-0610 was added on to ruxolitinib. And with the addition of CPI-0610, again, you can see on the right-hand side, a very nice decrease in the waterfall plots in the spleen volume and also an improvement in the total symptom score. And finally, when you look at this last, on the next slide, this last study done with CPI 0610, this is for patients who were on ruxolitinib, but instead of adding on CPI 0610 to ruxolitinib was discontinued and CPI 0610 was started. And again, what you see here is that even with single agent CPI 0610, you have very nice spleen response rates, and you also have very nice changes for the better and the total symptom score. So, you know, in my opinion, I think bromodomain inhibitors really are going to find a home in the treatment of patients with myelofibrosis with large spleens and a lot of symptoms. Next slide. And with that, I am happy to pass over the baton to my colleague from the Massachusetts General Hospital.
Hi, thank you so much. And after that whirlwind tour, which Dr. Stein got you through, what I am going to do is talk about myeloma. As usual, at ASH, there's always excitement with all heme malignancies, as you can see, and specifically multiple myeloma. What I think I will do first is talk about some of the trials which were presented, which are going to in fact impact practice and influence how we take care of our patients. And then we'll talk about all of the recent advances and excitement in the cell therapy space with bispecific T-cell engagers as well as ADCs, not just with BCMA this time, but for the first time at ASH, we are seeing different targets which are being considered as well in the bispecific space. So with that, the next slide, please. So the Apollo trial was presented. This is a phase 3 large randomized trial, and it is a trial using subcutaneous daratumumab. Subcutaneous daratumumab, as you all are familiar, was approved last year as a single agent. This is the first trial showing a combination approach with subcutaneous dara, and in this case, pomalidomide and dexamethasone. This was a large randomized trial after patients had received at least one prior line of treatment, but they had to have seen both an IMiD as well as a proteasome inhibitor, so lenalidomide as well as a PI. Relapsing following this, patients were randomized to receive either PomDex alone or pomalidomide dexamethasone with subcutaneous daratumumab. What is shown out here is the response rate, high response rates of close to 70% with the triplet as opposed to 48% with the doublet. And most importantly, what you are beginning to see even in the relapse refractory space, and this is a theme which is going to continue along, is MRD negative disease in patients with relapse refractory disease, with the triplet giving a 9% MRD negative disease. This obviously translated into a progression-free survival at 12-month median of 52% versus 35% for the doublet. If you look at the safety profile here, dara-PD did not have any unexpected toxicities. The big toxicity that we are all familiar with is myelosuppression, as well as a slight increase in infections. So this is going to become more and more used. In the US, it was already used. This Apollo trial is going to allow the use of dara-pom-dex even in Europe, which typically was not approved in that setting. Next slide, please. I did want to mention the Griffin trial because I know we've talked about triplets, but I think the future in myeloma already at the time of diagnosis is using quadruplets. This is the Griffin trial, and this was just an update of what was already presented at ASH last year and ASCO earlier this year. This is a phase 2 randomized trial, which is comparing RVD or lenalidomide bortezomib dex with the addition of daratumumab to this triplet. So a first quadruplet, and what you see out here is a transplant eligible patient population, 207 patients randomized, 104, 103 either arm, and this is a longer kind of a follow-up of this patient population. What is really important here to note is the MRD negative disease state in this patient population as you go from initial induction followed by consolidation towards maintenance. So at the cutoff of about 12 months of treatment, what you are seeing is nearly 80% of patients are becoming MRD negative in the quadruplet arm compared to about 40%. So a doubling of MRD negativity. And if you look at the response, it deepens over time, as you can see. Obviously, this translates into a remarkable progression-free survival as well as an overall survival, although no difference in the overall survival between the triplet and quadruplet. We do know that MRD negativity translates into improvement in overall survival as well. Next slide. Did want to mention another quadruplet. So sure, we're using quadruplets up front, but this was data which was presented this year's ASH. This is DARA-KPD, daratumumab, carfilzomib, pomalidomide, and dexamethasone in patients who are relapsed. Again, not a large study, but having said that, we are seeing very high response rates with about 55% of these patients becoming MRD negative if you use the quadruplet, and this again translating into a progression-free survival and an overall survival at 12 months of 84 months and 76 months at 2 years. So quite remarkable in the relapse refractory space, underscoring that we're really using both IMiDs, both PIs, as well as the CD38 monoclonal antibody in combination earlier on in the treatment paradigm for multiple myeloma. Next slide. But obviously all the excitement is in the cell therapy space, and we saw more data presented at this year's ASH. This is the CARTITUDE trial looking at autologous anti-BCMA CAR T cells. This is cilta-cel, or the Janssen product. These are patients who are pretty relapsed and refractory. They've had at least 4 lines of treatment. And I think all of you are familiar with the initial data, but this was a higher number of patients, close to 100 patients treated out here. The overall response rates is close to 100%, not quite 100%, but close to 100%. And the important thing to note is the with the efficacy, the toxicity is fairly manageable. CRS was noted in patients, but mostly grade 1 and 2, and grade 3 and 4 was hardly ever seen. This translated into a progression-free survival of about 76 months at 12 at a year. And patients who achieved a stringent CR or VGPR obviously did a little bit better. Again, the follow-up for the CARTITUDE trial is a little bit shorter. You're all familiar with the KARMA data, and our hope is that we're going to have a cellular T-cell product approved in the multiple myeloma space very soon. Next slide. Again, I think what was quite remarkable at this year's ASH was a first-time off-the-shelf CAR T-cell product. This was the Universal trial, which was presented by Sham Malakondi from MSKCC. This is an allogeneic anti-BCMA CAR T-cell product. Again, does not require manufacturing, so underscoring the fact that this is an off-the-shelf product. Again, early data in about 35 patients or so, but what I wanted to underscore here was the feasibility of an allogeneic CAR T-cell product with very high response rates once you get to a phase 2 dose level. And these patients, obviously, because this is an off-the-shelf product, do not require any kind of bridging therapy. They are required to be treated with ALLO-647, which is the homegrown allogene CD52 anti-CD52 monoclonal antibody to prevent graft versus host disease. Again, important to note that neurotoxicity, CRS, GVH, etc., was really very manageable with a very high response rate in this off-the-shelf CAR T-cell product. Next slide. we saw a lot of interest this time with bispecific antibodies. This is the Pfizer bispecific antibody, again, a phase 1 dose escalation trial here. And I think the theme with all BCMA bites, like the CAR T cells, is a very high response rates. The difference here being these are products which are off the shelf. They can be given to patients quite easily. They do not require any bridging therapy. And despite these patients being very, very heavily pretreated, we are seeing response rates once you get to the MTD of about 80% with this specific bispecific T cell engager. If you look at toxicity again, it is not trivial. They do need to be hospitalized for the first couple of doses, but most of the CRS is very manageable, and neurotoxicity was seen very minimally with this approach. Next slide. Similarly, we had other bites which were presented as well. This is the Regeneron BCMA CD3 bispecific antibody, again, a dose escalation trial. And again, if you look at the response rates, high response rates, and the duration of response with all of these bispecific T-cell engagers is at least 6 months. So patients are able to continue on these treatments with very manageable toxicities. Next. AMG 701 is the Amgen BCMA bispecific T-cell engager. Again, this was a phase 1 dose escalation trial. The response to date, again, we are seeing stringent CRs as well as MRD negative disease in this patient population. Again, toxicity is manageable when you use these bispecific T-cell engagers, and we do have patients who are able to stay on drug if they are responding for protracted periods of time. So the bispecific T-cell engager space is going to be really exciting, and we will have to figure out how we're going to use or adopt this in combinations in the future. Next slide. In addition to teclistamab is the last one. This is again another BCMA bite from Janssen. I'm not going to spend too much time on this, but the theme is the same. Again, high response rates, as you can see, close to 73% with this one, and complete remissions as well as stringent complete remissions. And the important thing, as you can see in the duration of response, is patients are able to stay on drug for well beyond a year if they are actually responding. Next slide. Now, in addition to all the BCMA, whatever I've presented so far was all in the BCMA space. At this year's ASH, we had 2 other targets which were also shown, and this is talquetamab. This is a bispecific T-cell engager against GPRC5D. This is a G protein coupled receptor. We don't completely understand the function of GPRC5D, but nonetheless, it is expressed on multiple myeloma cells. And what was really interesting to us is once you get to the MTD with this bite again, you're seeing a response rate of 73%. Again, in my mind, extremely important. Specifically, we are going to be in a space where we are going to be treated with an anti-BCMA strategy, and then what do we have to help in that refractory space? So talquetamab seems to be a really good drug in this space, and again, showing a lot of durability of response as well. Next slide. Similarly, we have another bispecific T-cell engager. This is against FcRH5. Again, one doesn't really completely understand the function of FcRH5. Nonetheless, it is overexpressed on multiple myeloma cells. And once you get to the MTD, you're seeing an overall response rate of above 60% in this patient population, with durable responses in people who are responding well beyond a year using the BFCR 4350A bispecific T-cell engager. So really exciting data at this year's ASH, not only in the BCMA space, but for the first time now we have 2 other targets which are showing high response rates with stringent CRs in a relapse refractory multiple myeloma space. Next slide. So with that, I will hand it over to my colleague who's going to go over lymphomas, Dr. Matthew Madisar. Thank you.
Thanks, Dr. Raji. That's a tough act to follow, guys, but there is definitely a lot to say about what's going on currently in lymphoma, certainly more than we can get through here together, but I'm going to try to touch on some key themes and certainly continuing on and building on the themes that Drs. Stein and Raji have already highlighted for us today. Next slide. So bispecifics, you know, Dr. Raji has talked a lot about the progress we made in myeloma, but let's be honest, lymphoma started this. And mosunetuzumab is the bispecific that's furthest along in clinical development, both in the management of aggressive and indolent B-cell lymphomas. Shown here are some proof of principle data from Dr. Olszewski and colleagues who attempted to use mosunetuzumab as monotherapy and as first-line therapy for newly diagnosed large cell lymphoma in patients who were extreme elderly, 80 plus, or 70 and above, 60 and above and unfit for anthracycline-based chemotherapy. It was given as in its intravenous formulation according to the regular schedule with dose escalation during the first cycle with ramp-up dosing, and then Q3 up to a year of treatment. We see that there is definite T-cell activation happening in newly diagnosed patients, both CD4 and CD8, as well as appropriate release of interferon gamma. The response here, I would say, is interesting, although far from a home run. We certainly did see complete responses in the data that Adam presented, although certainly not universally so, and there were progressions and deaths on treatment. But it's a reasonable proof of concept that in frail patients, you can administer mosunetuzumab monotherapy in large cell lymphoma, even without prior exposure to anthracycline-based chemotherapy, and see responses. Interestingly, although not highlighted on this slide, there was no grade 2+ CRS in these patients, which was a surprise to the investigators, given that patients went on study with a moderate burden of disease. Next slide. Perhaps a more appropriate and more sustainable model here is the pilot data that was presented for MCHOP. This is mosunetuzumab combined with CHOP chemotherapy. For those not familiar, mosunetuzumab cannot be co-administered with rituximab. They have competitive binding. That's why we're all missing the Rituxan here. That being said, again, it was given according to its regular schedule. In this study, mosunetuzumab was administered subcutaneously, and it is being developed both as an IV and sub-Q formulation in parallel by Genentech. There's still relatively few patients treated here. The majority of patients were first-line large cell lymphoma. There were a handful of relapsed refractory patients treated. But in the upfront study, we see that there were good responses with a high overall response and high complete response rate. You will see on the table on the bottom right and with the dark blue heading that there were patients who did come off due to toxicity, and there were patients who had progression of disease. There were 3 patients who had POD on MCHOP chemotherapy out of those treated. It's a relatively small number, but 3 PODs is a little bit of an unusual number to see. It may just be bad luck, or maybe telling us something about this combination that's shown in the bottom left here. There remains a fair amount of uncertainty in the field whether combining a bispecific that's relying on T-cell engagement with chemotherapy, including prednisone, which is modestly T-cell toxic, whether concurrent administration is really the best approach forward, or whether we should be developing concurrent schedules with either an induction or consolidation/maintenance with a bispecific. That being said, the field is moving ahead with planning concurrent combination trials in a number of settings, combining a bispecific antibody with chemotherapy, perhaps without a lot of underlying scientific data to support this progression of research. Next slide. Here's data in mosunetuzumab monotherapy in relapsed refractory follicular lymphoma here with the intravenous formulation. This is what is going to lead to what we expect will be the first FDA indication for a bispecific antibody, that being in relapsed refractory follicular lymphoma, where we see very high overall response rates, very high complete response rates, and quite excellent durability of response. We see responses in FL even in our highest risk patient populations highlighted on the bottom left here, patients who were refractory to the prior line of therapy, so-called double refractory, both to rituximab as well as alkylating-based chemotherapy. The POD24 patients, and that story continues to evolve. Those refractory to prior PI3K, and those who had received prior CAR-T, although that was a very small number of patients. When you look at the T cell activation, what we do see is that there is sustained activation of T cells throughout the course of therapy, at least up to cycle 8. We're still learning about the durability beyond cycle 8 for those who had partial response. In terms of toxicity, there is relatively low rates of severe CRS highlighted on the bottom right here. Grade 2 CRS is uncommon with mosunetuzumab, less common than with Genentech's other antibody, glofitamab, or as we will see in the subsequent slides, other bispecific antibodies being developed by competing companies. Next slide. Speaking of which, here's data for epcoritamab. This is the molecule being developed by AstraZeneca and Genmab, the so-called dual-body antibody. And here are some preliminary data from the phase 1/2 that were presented at ASH by Dr. Hutchings. Unlike we saw with mosunetuzumab in the last slide, there is a greater degree of grade 2 CRS. This may be more acceptable in the myeloma patients. Dr. Rajay said, oh, this is good. We only have grade 1, grade 2. At least we're not seeing grade 3+. In the lymphoma space, we're hoping to find a bispecific antibody that really limits to grade 1 in the majority of patients, because in our world, grade 1 versus grade 2 is what defines whether you need to be admitted for observation and management, and thus consuming hospital-based resources that currently in the COVID era are increasingly scarce. So EPCO did have a relatively high rate of grade 2 CRS in the reported patients. In terms of activity, we do see anti-tumor activity in both aggressive and indolent B-cell lymphoma. Relatively fewer patients with indolent lymphoma treated, but response rates were nearly ubiquitous. In large cell lymphoma, we see both PRs and CRs, particularly in those patients who are receiving treatment at therapeutic dose. We saw 10 of 11 patients treated at the higher doses of EPCO achieving overall response. It's a very nice number. Next slide. Odronextamab, this is the Regeneron bispecific, and here we have phase 1 data that were presented at ASH this year. A smaller number of patients that were reported on. Every company has its own strategies to try to mitigate CRS. Regeneron's trying to split the dose in a step-up dose fashion where they're giving it over 2 days, really trying to approach it gently. Despite these mitigation strategies, CRS was still a problem. We see that there was a little bit of grade 3 CRS in treated patients and even one case of grade 4. In terms of the grade 1/2, grade 2 CRS is still quite common despite the mitigation strategies that Regeneron has deployed to date. Responses in follicular lymphoma are excellent. We see that the majority of patients had a measurable response and everybody had some decrease in their tumor burden. In the large cell patients, there were fewer reported, but there were responses seen. The durability of response, particularly in the FL, leaves a little bit to be wanted. It's a little bit undesirable with a drop-off really by 12 months. We don't have the full pharmacokinetic data available from Regeneron yet, but one wonders whether this is being juxtaposed against the higher rates of CRS, whether what this antibody is achieving is a more rapid activation, demargination, and expansion of T cells in vivo, leading to earlier exhaustion, which may correlate with loss of durability. It's the slow burn versus fast burn hypothesis that we see in CAR-T products. It may be what's at play here, although we certainly are hoping to learn more in the months to come. Next slide. Moving away from bispecifics, here's data from VLS-101. This is an antibody drug conjugate targeting ROR1, which is an antigen that's relatively ubiquitously expressed across B-cell lymphomas as well as on solid tumors, which is certainly beyond any of our purview today, but I think the company's excited about it. The payload here is MMAE, which is the same payload from polatuzumab vedotin, which already is, of course, an antibody drug conjugate that's FDA approved in the treatment of multiple relapse refractory large cell lymphoma. It's unclear to me whether the ROR1 target has advantages over CD79B, which is the target for POLA. That being said, we do see activity with this payload targeting ROR1 in both large cell lymphoma, relatively few patients, as well as in mantle cell lymphoma, where there were more patients that were reported by Dr. Wong out of MD Anderson this year. Interestingly, Genentech really hasn't had a very aggressive developmental program looking at POLA and mantle cell lymphoma. So this may create sort of an ecosystem in which another MMA delivering ADC could have a pathway forward in the management of lymphoma. And we look forward to seeing a greater number of patients treated and to learn more about the durability of response in those patients. In terms of toxicity, the ROR1 seems to not be expressed on healthy tissues, and the toxicity reported in this early experience was really that which we're accustomed to seeing with MMA deliverers, myelosuppression, and neuropathy. Next slide. Perhaps making a big splash in both the leukemia world as well as lymphoma is the LOXO non-covalent BTK inhibitor. And these are data that were presented by my friend and colleague, Anthony Mato, showing the use of LOXO in a relatively heavily pretreated population of CLL. The vast majority of patients had either previously received or been refractory to prior BTK, either as monotherapy or in combination approaches. And as shown on the bottom left, the box and whiskers show you that prior exposure to BTK inhibitors really did not lead to resistance to this inhibitor. And we really do believe that LOXO 305 is going to be active in BTK resistant or refractory patients. The waterfall plot shows you that there is nearly ubiquitous activity in these patients in terms of tumor reduction. And while there's relatively few patients that are further out, you see the number at risk in the bottom right really quite small beyond 8, 10, 12 months. Nonetheless, there does seem to be some durability of response in these early experiences. And we look forward to seeing these data mature over the year to come. But this agent definitely has a lot of enthusiasm in the leukemia and lymphoma world, either following BTK or potentially improving on currently covalently linking BTK inhibitors. Next slide. Here's some data from TG presented in their sort of U2 program, their umbralisib, which is an oral PI3K/CK1 epsilon inhibitor combined with their homemade anti-CD20 antibody ublituximab. It's paired against good old G plus chlorambucil in either treatment naive or relapsed refractory CLL. And while many of us are growing tired of seeing G chlorambucil trotted out as a comparator arm, we do see that U2 does beat G chlor in CLL in terms of progression-free survival in treatment naive as well as relapsed refractory patients. Toxicity-wise, it does seem like there was a greater frequency of grade 3+ AEs in the U2 arm than we've seen in earlier studies. The company continues to tout a tolerability profile, including a lower rate of the sort of the oral PI3K triad of hepatitis, pneumonitis, enteritis, although we're waiting for more granular data from this experience. The PFS curves here are a little bit, in my opinion, disingenuous because obviously G-chlor stops after the first 6 cycles, whereas umbralisib is continued until progression or intolerance. But a median progression-free survival of approximately 3 years is attractive. And once we know more about the toxicity profile here, this program could have a way forward in terms of registrational approval. Next slide. CAR T-cell therapy and certainly cellular therapy is very much du jour in aggressive lymphomas. The question is whether this has an approach and a life to it in the treatment of indolent B-cell lymphoma. ZUMA-5 is an attempt at answering that question. Karen Jacobson presented the data on 146 treated patients, the vast majority of whom had follicular lymphoma. There were a smattering of MZLs, both nodal and extrasomal, that were included. The data for MZL here are both less robust and less interesting. Sorry about that. In terms of follicular lymphoma, there is definitely a very high overall response rate. And a very high complete response rate. The durability here is still relatively immature, but this does appear to have some durability in terms of progression-free and certainly overall survival. The majority of those durable responses did appear to be those that achieved a complete response. PRs seem to drop off quite quickly, which is not surprising. The question for us in the community, of course, is whether, given the challenges of administering CAR T-cell therapy, both in terms of logistics as well as the financial toxicities, whether this really is superior to off-the-shelf immunotherapy such as biospecifics, which we expect to get registrational approval, My opinion is that given the tolerability and activity of drugs like mosunetuzumab, that it's going to be harder to justify all of the challenges that are associated with administration of autologous CAR T-cell therapy in indolent lymphoma. But we'll see how these data mature and whether there is a value proposition to be found using CAR T in these disease states. Next slide. I think that's my last. Thank you all for your attention. I think we move into the more interesting part now, which is discussion.
Yeah, so we have a few questions on the chat. If we want to take some now, or do we want to move all of those to the end? Let me know. We have some questions on lymphoma, myeloma, AML, pretty much across the board.
We're ready. We're ready for the questions.
Should I start? So if you see the Q&A panel, you could see you can see the questions there, and I've added a few to the chat as well. But I'll read out the first one, which is essentially on. Can you speak to the long-term thinking about Ven plus AZA as backbone in AML and MDS? That's for Dr. Stein.
Yeah, sure. So, we didn't talk about this, but venetoclax and azacitidine has certainly become the standard of care for patients, older patients with acute myeloid leukemia, who cannot get intensive induction chemotherapy. Now, my feeling about this is that we're already using Ven plus AZA, not strictly according to what the FDA label said, which is only for patients 75 and older, or patients younger than 75 with comorbid medical conditions. I think many of us are using Ven plus AZA in patients over the age of 70, over the age of 65. And I really think that over the next 5 years, there's going to be a big effort to replace induction chemotherapy with 7+3 with chemotherapy or induction therapy with Ven plus AZA. So I think that Ven plus AZA is going to end up being the standard backbone 5 years from now. Perhaps the exception to that will be in patients with favorable risk core binding factor acute myeloid leukemia, where we know that that intensive chemotherapy actually can be curative. But I do think Ven AZA is the wave of the future.
Got it. The next question is for Dr. Mattisar. Is there a difference between these CD20, CD3 agents at this point?
That is the $64,000 question. We're all trying to answer that ourselves as we choose which studies to move forward. I think that the answer is yes, although we have an incomplete picture as to how they precisely do differ. But we all know that these drugs do differ in terms of their toxicity and their activity. And the general sense that we have is that the ones that are more toxic are a little bit more active. And we're trying to find that sweet spot where we have an antibody that is active enough, but well tolerated enough that it can be administered, particularly in a community-based setting. This may differ, and the sort of risk-benefit profile may differ in disease states. In large cell lymphoma, where options are fewer and risk of death due to progression is greater, we may accept a more toxic bispecific if it's going to get us where we need to be in terms of disease control. Whereas in indolent lymphomas, where risk is lower, options are more numerous, we may prefer a slightly less active antibody if it can be administered safely in an outpatient setting in a community setting.
I guess the last question for Dr. Raje before we move to the next session, and then we can continue with the Q&A towards the end. What is the future of cell therapy in myeloma? What are your views on NK cell-based therapies?
Yeah, all good questions, Wajiha. I do think cell therapy is going to get approved. At least the first drug product we should have in March, and then we hopefully we'll have 2 drug products soon. So I do think we're going to be using more and more of cell therapy. And, you know, the data we have right now is in sort of end stage myeloma, wherein we are seeing extremely high response rates. And the durability is obviously, you know, we're seeing if you achieve a CR or a stringent CR on a median, the PFS is about 18 months. Now that is remarkable in this patient population. There are ongoing trials with all of these different drug products wherein we are bringing it up earlier in the treatment paradigm, and we'll have to see whether or not we can really translate the durability of response into more longer lasting durability. So that is here to stay. In terms of your question, was it bispecifics that you were talking about after that? So,
NK
or
cells.
NK cells, I'm sorry. NK cells, very early. The problem with NK cell CARs, as you all are familiar, you know, NK cells don't stick around. The benefit of NK cells may be that you can create an off-the-shelf product again, but we already have other allo products as we presented at this meeting. So I think NK cells, we need a lot more work to figure out where they will kind of fit into the treatment paradigm with myeloma.
Thank you. We have one follow-up question for Dr. Stein. It goes like, when AZA, as you indicated, is the future backbone chemo, can you comment if when decitabine is equal to when AZA?
Sure. So the randomized phase 3 Viali-A study was done with azacitidine venetoclax versus azacitidine. However, the FDA label approves the use of any hypomethylating agent. The only other one out there is decitabine with venetoclax. So I think the feeling in the community is that, or among academic physicians, is that they are likely equivalent. Let me make one more comment that decitabine actually has a leg up in certain ways. And that's because there is now an oral formulation of decitabine, and there is an ongoing clinical study looking at whether the oral formulation of decitabine with venetoclax, which is oral, achieves the same levels or PK of both ven and decitabine as it would with IV decitabine and venetoclax, meaning that it may be that decitabine venetoclax will be an all oral regimen that will be very attractive both to physicians and patients, and therefore perhaps it will supplant IV azacitidine with oral venetoclax.
Thank you. So in the interest of time, we'll move on to our next section, which is what to expect in 2021 from the different heme malignancy space. Some of the previous slide, please. Yeah. Some of the discussions that we were having internally were on the new trial readouts, some expected data, new trials which will be initiated, some approvals as Dr. Rajay mentioned on the cell therapy side. So perhaps 5 minutes or so of your perspectives, starting from Dr. Stein, on what do we expect in 2021? What's the exciting data that we should look forward to?
Yeah, so I think, you know, this kind of data, at least in myeloid malignancies, goes in cycles. So over the past 2 to 3 years, we saw some big name drug approvals. And I think now we're sort of starting the clinical trials that may lead to more drug approvals in 2 to 3 years from now. The trials that are exciting are those trials that are sort of like, we're a little bit behind myeloma, but now we're starting with triplets. So now we're starting with AZA venetoclax, where we're adding in targeted therapies in an effort to improve both response rates, but maybe more importantly, improve the event-free and the overall survival. So we've already seen at ASH this year, something we didn't talk about, are triplets with azacitidine venetoclax and the FLT3 inhibitor gilteritinib. There was data presented at ASCO last year of azacitidine venetoclax and the IDH inhibitor ivosidenib. There is a clinical study that is getting going that's going to be looking at azacitidine venetoclax and the RAR alpha agonist SY1425. So all of those early studies, the early phase 1-2 studies, we're going to see readouts of those probably by ASH 2021 or ASCO 2022. I think in the field of myelodysplastic syndromes, that's where things may be a little bit more open and perhaps a little bit more exciting. You know, in high-risk MDS, there's a randomized placebo-controlled phase 3 study of magrolimab, that anti-CD47 antibody, with AZA versus AZA monotherapy, with the primary outcome in that study actually being the rates of complete remission. That's a study that probably won't read out this year, but what will likely read out in 2022. And that really could change the standard of care in high-risk MDS. I guess the final study that I'd point out is that, you know, we've seen a lot of data, as we just talked about, with AZA venetoclax and acute myeloid leukemia. For myelodysplastic syndromes, there's also very good data with AZA venetoclax, and a randomized phase 3 study of AZA-ven versus AZA is planned for patients with high-risk myelodysplastic syndrome. One other thing I want to say is that everyone always asks me about, you know, you hear about in lymphoma and myeloma all these CAR T-cell therapies and how well they work, and I'm quite jealous of my colleagues that they have these tools. You know, in myeloid malignancies, I don't see cellular therapies like CAR T-cells really playing a major role in the next few years. And the reason is because the targets in myeloid malignancies are not nearly as good as they are in myeloma and in lymphoma, right? So when the targets that we have in myeloid malignancies tend to myeloablate people, and after you myeloablate someone, you've got to transplant them. And that becomes much, much more complicated than giving a CAR T-cell where patients' counts then recover and, you know, they're in a complete remission.
Dr. Raji?
Yeah, so, you know, along the same lines as what Dr. Stein has just mentioned, I've already said this, a lot of excitement. I think the big thing which is going to happen this year are twofold. We're going to have 2 CAR T-cell products which are going to be approved. Obviously, the approval is going to come in the late stage wherein patients are completely refractory. We expect ide-cel to get approved, and then our hope is that cilta-cel gets approved shortly thereafter. So we're going to have a choice of 2 CAR products in the relapsed refractory multiple myeloma space. And the question is going to be who uses what or which patients, because I do think the eligibility for both of those CARs is going to be very similar. There's a lot of patients waiting for these drug products, so my sense is that how we use these is largely going to depend on what's available, the manufacturing, access to manufacturing, and who can get the drug product to them, because in terms of costing, I'm pretty sure that it's going to be, or it will need to be sort of comparable between these 2 cellular products. I think what you're going to see end of the year is more data in specific subpopulations with these CAR T products, that is specifically in the high-risk patient population. Can we go in earlier, and can we actually change the natural history of these diseases? Or can we even consider replacing an autologous transplant in a subset of patients? And I think the focus will be on high-risk patients. These are ongoing trials already, and the hope is that at the end of the year, we will have a little more mature data to speak to bringing these cellular products earlier on. I do want to mention testing also, MRD testing. We've talked a lot about this. You saw data in the upfront setting where we are getting to very high MRD levels. We now have a test which is an approved test by the FDA. It is a genotypic way of looking for MRD. And I think using this in future studies where we are seeing extremely high response rates as a surrogate to translating into a PFS benefit is something we're all going to hopefully use. I do think the FDA is very open to that idea as long as we design the trials in an appropriate way, looking at MRD as an early endpoint to get a sense of for drug approval also. I think the paradigm of treatment is going to change over the course of the year. I've already shown you 4 drug combinations in the upfront setting as well as in the relapse setting. So most patients are going to be exposed to a proteasome inhibitor, an IMiD, and a CD38 monoclonal antibody, and certainly all of them by their second relapse. And so the whole BCMA space is going to open up earlier and earlier. I think the BiTEs are an exciting space to look out for, but they are going to be used probably second-line treatment or third-line treatment. And I think our challenge over the next 2 or 3 years is going to be in, we have a lot of drug products in the BCMA space. Is there a potential to combine them, to sequence them? These are questions which have not been answered as yet. And I do think we're going to be doing trials to try and address some of these. And lastly, I will mention that, you're going to have very quickly in a couple of years, the post-BCMA space. So people who are relapsing after a BCMA drug product. And most of the data to date suggests that you still have BCMA expression. So it's not because of lack of expression of BCMA, which is the cause of resistance in the majority of these patients. So can you sequence some of these BCMA targets? And if you've lost BCMA, we've already talked about 2 bispecific T-cell engagers targeting different antigens on the tumor cell. And I think those are going to be what's going to be used. The combination approaches with these BiTEs, with IMiDs, with proteasome inhibitors, or with even with CD38 monoclonal antibodies are ongoing studies, and we'll see more data to speak to that as well.
Thank you, Dr. Raji. Dr. Madhusar, and probably the first question to you is that will the bispecifics make it to the market in 2021?
Yeah, I think we all expect Mosun to get approved for FL, relapsed FL in 2021. I think we're probably going to see 4-ish new approvals this year in lymphoma. We'll see Mosun, we'll see loncastuximab for relapsed refractory large cell lymphoma. We'll see liso-cel get its approval finally as the 3rd cellular therapy product for large cell. And I expect that we'll see a new approval for copanlisib plus rituximab in the treatment of indolent B-cell lymphomas, probably across the indolent spectrum. So those will be our major approval advancements this year. And then we'll see ongoing studies in CLL. We'll learn more about LOXO. We'll learn more about combination novel, and particularly with studies with MRD as an endpoint and trying to create programs that move away from the paradigm of progression to intolerance or continuing until progression or intolerance, and moving towards a time-limited MRD-guided approach in the treatment of CLL. Mantle cell lymphoma, I think, continues to be an area where we're not going to see a lot of major progress this year. We have studies that are in early development, but the only practice-changing study that's ongoing is Simpatico, which is looking at ibrutinib plus venetoclax, and we probably won't get a readout of that until 2022 at the earliest. Then, of course, in large cell lymphoma, the study that we're all awaiting readout for so eagerly is POLARIX, which is the randomized trial of R-CHOP versus R-CHP plus polatuzumab vedotin in newly diagnosed large cell lymphoma. We'll get readout of that June, July of 2021. And certainly if that's a positive study, that will have practice-changing implications.
Thank you, Dr. Madhusar. Next slide, please. The COVID-19 pandemic continues to rage in 2021 as well, and the impact on cancer patients across malignancies has been tremendous. So just want to get your sense of how you see care evolve, considering the scenario that probably the role of vaccination will play out. So just want to get your sense on how the heme patients have been impacted, and how do you see them in 2021 being treated in the COVID scenario? Dr. Stein.
We're all politely waiting for our colleagues to speak first.
Did you call on me?
Yes, please.
Okay. Well, I can say in the context of myeloid malignancies, I think initially, at least, it accelerated the move to AZA venetoclax because, rightly or wrongly, there was a feeling that, well, there were two things that happened. One is at the beginning of the pandemic, I think there was a feeling that maybe a hospital wasn't a safe place to be because of nosocomial spread of COVID. And secondly, we were instructed, many of us by our institutions, to do as much treatment outpatient as possible to leave room for the COVID patients inpatient. And that therefore makes us use outpatient regimens like AZA venetoclax much more than an inpatient regimen like induction chemotherapy. And I think with the increasing familiarity both academics and community doctors have with AZA venetoclax, that's accelerating, or hypomethylating agent venetoclax, if you include decitabine, I think that's accelerating the what I talked about previously, and that is the use of this combination beyond what is in the FDA label.
I think I'll just go next, Wajiha, for myeloma. Very similar to what Dr. Stein said. The initial instruction was try and keep people home. I think what we have learned with the pandemic is there's a few things that we can do quite well with patients being at home. We've all transitioned to using telemedicine and televisits. And in a disease like myeloma, where a lot of oral drugs are used, that's been very effective, actually, specifically in patients who are on maintenance treatments. I think the biggest thing that we have learned, and this is really, really important for most to understand, is the best thing that we can do for our patients in terms of their outcomes is make sure that they are in a good remission. So whatever they require for us to get them into a remission is critical, because we've seen outcomes across the globe. And as an International Myeloma Society, we've actually collected data on patients. And if a myeloma patient is in remission, their outcome with COVID is going to be much better than a patient who is not in a remission. So I do not think we should, compromise the care of the patient. And I will argue and say the hospital is probably the safest place for a patient right now. We have not seen nosocomial spread. The issue, though, is how the hospital resources have been stretched. They've been stretched to a point where we've not been able to for example, my whole unit, the lymphoma myeloma unit, was transitioned into early on in March and April into a COVID ICU. Thankfully, that's not happening right now, although the numbers are going up quite high. But if a patient requires treatment for their myeloma, they should not procrastinate, nor should the treating physician procrastinate. The good news with myeloma, we have oral as well as regimens, which can be quite easily given in the clinic. What we've also done, at least at our institution, is used our affiliates, and we've tried to decentralize our myeloma care so that not everything has to happen at MGH, but can happen at our affiliate site in Danvers or Waltham, and that's been really helpful to patients. In terms of clinical trials, we did make some adjustments. Obviously, we're not going to be doing sort of a new drug, which is a first in human sort of study with very little track record. So we put that on a back burner. But clinical trials which have a real impact, such as these bispecific T-cell engagers, such as CAR T-cells, we as an institution decided to continue with. We continue to accrue patients on those clinical trials. And one of the thinking behind this was we obviously needed COVID testing on them, but we did keep all CAR T-cell as well as bispecific T-cell engager trials open, with the idea of saying that if you can get a patient in remission, their outcome is probably going to be the best. In terms of vaccination, we are going to have recommendations. Our recommendation for all myeloma patients would be to get the vaccine when available. The issue is going to be how robust a response to the vaccination, and that's data we don't have, but we hopefully will be collecting antibody data on our patients as well. The vaccine is certainly not going to do any harm to our patients. How much it helps would depend on immune paresis of the patients and depends on where they are in their treatment course.
Thank you, Dr. Raj.
In the lymphoma space, I'd say that there's some sort of general trends and then there's specific issues. The general ones are that we've seen a movement away from patients being treated in academic centers that are at a distance from their primary residence. And patients are more likely to be receiving treatment closer to home. That's a shift towards private practice. That's a shift towards regional or network setting, as Dr. Rajay alluded to. That has implications in terms of the overall progress of clinical research, as well as the survivability of major academic medical centers, which are facing a financial pinch due to these changing demographics. Whether they're short or long-term remains to be seen. In terms of specific lymphoma matters, we certainly have, as a community, changed how we're treating patients. During times of surge, when we expect our hospital resources to become more scarce, we curtail hospital-based activities, including inpatient chemotherapy, including CAR T-cell, including stem cell transplantation. When we're not in a resource-constrained setting, we still do alter how we practice, and many of us are choosing not to administer maintenance therapies like maintenance rituximab, which may be associated with progression-free but not overall survival benefits, due to a belief that administration of rituximab may dampen the response to COVID vaccination when it becomes available to our patients more broadly. Questions regarding the activity of the vaccine in a number of clinical settings are going to need to be answered in a timely fashion, including patients that are on chronic oral therapy with BTK or PI3K inhibitors, as well as patients who are receiving required treatment that includes monoclonal antibodies or other lymphodepleting therapies, so that we can have better guidance for ourselves and for our patients about how to approach vaccination in a setting that it continues to be resource scarce.
Thank you, Dr. Matusar. With that, we'll move to the final section of our town hall, which is a Q&A. We'll continue on with the questions that have been posted onto the chat and Q&A. I request all attendees to continue posting the questions, and I, from my end as well, will keep adding. Let's look at some of them. Question for Dr. Stein. For APR 246, what do you make of their press release post-ASH saying that there is no statistical improvement?
Yeah, so I mean, in addition to that it was negative when it came to statistically significant improvement in CR rate, they also said there was no statistically significant improvement in overall survival. That's not surprising, given that the CR rate wasn't statistically significant. I mean, I think that Apria has a real challenge on their hands. I mean, I think that there had been skepticism previously that APR worked as a p53 targeting agent. And given the negative MDS study, it raises the question why this compound would work in any other p53 mutant clinical setting. So I think they have their work cut out for them to really try to pin down whether this is really a p53 targeting agent, and if it isn't, is there a path forward to use it more generally in patients with myeloid malignancies?
Thank you, Dr. Stein. Question for Dr. Raje. What is the current thinking about the use of MRD data as a surrogate from a regulatory perspective in myeloma?
So good question, Wajiha. As I mentioned earlier, we do have an approved MRD test through Adaptive. It's genotypic testing. And I think the key is we do have data to also show that MRD does serve as a surrogate, not just for PFS, but also for overall survival. So as long as MRD testing, and it doesn't matter which one you use, we have flow-based 10-color flow from Europe, as well as the genotypic one, which we tend to use more of, available. As long as the trial incorporates it at specific time points, I do think it could be used as an early readout. It is going to need confirmatory PFS data. You cannot use MRD as a standalone test, but the FDA and the regulatory bodies do have an appetite to want to use these early endpoints to see which way trials will be going. So I do think it's here to stay, and I think we have to start incorporating them in clinical trial design going forward.
Thank you so much. I think the next two related questions are probably for all the three panelists. From an industry and the new drug launches perspective, in view of COVID, how do you see shared decision making, especially for new products that are being launched in 2021? Also, given the pandemic where access to industry sales and medical representatives is limited, how do you see drug launches being impacted? These two related questions, and probably more relevant for lymphoma and myeloma, where we will see some new modalities as well as therapies.
Yeah, I can get started if you want, Wajiha. You know, this is a challenge, and it's a challenge all around. Certainly for us, when we've had two-cell therapy products, and the rules around COVID really have created issues because they could not come into the institution for monitoring, etc. But I think the nice thing was working collaboratively with industry, we were able to pivot into remote monitoring as well. So industry was also able to make changes in how a study is conducted. But you know, it's really important to note that, you know, there are some other drugs such as you know, we haven't even talked about them. Melflufen, for example, selinexor, for example, those have certainly taken a backstage. And we've also prioritized drug products such as the bispecific T-cell engagers and CARs. And we've made certain changes in our clinical trial protocol offices as to how we monitor, how we gather data. And it's been a process for all of us, I think.
I'd agree with that perspective broadly, and I think that the challenges here in terms of shared decision making regarding research is a recognition that, at least for a measure of time, clinical trial participants may be a little bit more scarce. So centers are going to need to be a little bit more judicious about their research portfolios and deciding how many studies to open, which studies are likely to accrue in this current climate, and how to prioritize programmatically what they hope to accomplish with and for their patients.
Thank you, Dr. Mattisar. Any insights from you, Dr. Stein?
I would sort of echo what my colleagues have said. Specifically, what Dr. Mattisar said about being more judicious in which trials get open. You know, specifically in the AML realm, everyone now is doing a triplet like we talked about before, but not, we can't open all those triplet studies. And it's also going to be important to try to figure out how to engage the community where many of these patients are now being seen into participating in these clinical studies.
Also, beyond the clinical studies, if any perspectives on new launches, and Dr. Raja alluded that some products have taken a backseat, which were launched very close to the pandemic or in the middle of pandemic. How any insights for how engagement with perhaps the industry colleagues will take place when these new drugs come into the market?
So the biggest thing that we are waiting for in myeloma, as you know, are the cell therapy products. As I mentioned, and we've had to work very closely with folks. The problem is going to be these are things which cannot be done out in the community. They have to be done at MGH. Even on the clinical trial, before we took a patient for CAR T cell, we had to make sure. There was a time when we were using tocilizumab for COVID patients. We're not doing that anymore. Those trials did not sort of pan out. But even before hospitalization, we had to ensure that we had enough tocilizumab and we had an ICU bed. Whether a patient goes to the ICU or not, once you are subjecting a patient who treatment, you have to ensure that you're going to be able to get them through. So these were high-level discussions we've had to have. Depending on how this pandemic evolves over the next, I think over the next year is what we are going to look at, and look at our ICU and inpatient volumes. We might have to roll out these drug products kind of slowly and strategically, and only use them for patients who absolutely need them. So the rollout is absolutely going to be not what we would have expected in a non-pandemic period, but that's where I think we have to work closely with our pharma colleagues on trying to ensure that we can do it safely, but it's going to be a slow rollout. There's no question about it.
Thank you, Dr. Raji. Dr. Mattis, I'm just curious, is that perhaps going to result in a slightly opposite shift in lymphoma, where the movement is a little bit from cell therapy towards bispecifics, or do you see any other ways in which the industry can collaborate to ensure that the correct patients receive?
So those are separate questions. So in terms of cells versus bispecifics, certainly there's a value proposition for anything that can be administered safely in an outpatient setting right now. I don't expect to see a bispecific approved for large cell in 2021. It's just not quite there yet. Maybe towards the end of the year if we're lucky. So I don't see that practice pattern changing much in the near term. The bigger question of how do you roll out a drug when everyone's on lockdown, and the traditional mechanism of drug rollout, which is sending out an army of drug reps to private practice offices to engage community oncologists, is no longer practicable. It's interesting. I think for academic settings, it'll be seamless. We've all transitioned to a virtual platform. Well, number of virtual ad boards that has largely replaced live ad boards successfully. So I don't see a challenge there, but there is going to be a burden on companies on how they're going to be able to successfully educate and message community oncologists in a way that's fundamentally different than how it's been done over the last 15 years. And that's going to be a challenge of how to use these digital platforms that we're all increasingly comfortable with in a way that is impactful and can actually reach the community oncologists that are the ones usually responsible for prescribing new medications.
Thank you, Dr. Madisar. Shifting gears back to some clinical questions. Question for Dr. Stein. Will STAMP inhibitor be used upfront in CML?
It's a good question. I mean, I think that from a clinical perspective, there's really no reason to use it upfront in CML. I mean, the current drugs that we have imatinib and dasatinib and nilotinib, all those drugs work really very, very well. Now, from a company perspective, certainly I'm sure they would like to move it up into earlier lines of therapy, and you may see clinical studies that test the proposition whether the depth of response in patients who receive a STAMP inhibitor is better than the depth of response in patients who receive something like dasatinib. I mean, at the end of the day, you can't show a survival difference because the survival is so good in patients who get even first generation tyrosine kinase inhibitors. So the clinical studies have to be based on something else. But from a clinical perspective, I personally don't really see a reason that it should be moved up into earlier lines of therapy.
Thank you, Dr. Stein. The next question is for Dr. Raje. How important is off-the-shelf benefit? Will there be an acceptance of slightly lower survival data just because of the sheer convenience of having either an off-the-shelf CAR-T or a bispecific?
I think having an off-the-shelf approach in myeloma is important only because myeloma is an immune disease. The fact that we've, with an autologous product, got such amazing data to me is a little bit of a surprise. My sense with the autologous or off-the-shelf products is going to be, are they going to persist? And for persistence, you need more of an autologous, which is why I talked more about, we are going to need to have a combined approach with some of these strategies. So we are going to use mix and match. The one thing with cell therapy products for myeloma patients, those patients who've had a CAR T cell absolutely love it because it's the first time in their lives that they are on no treatment whatsoever post a CAR T cell, and they've stayed in remission for years. So the persistence of the CAR T cell, which generally comes with an autologous, may be at the cost of a response, and therefore using an allo product followed by an auto, an off-the-shelf product followed by an auto product would be something we should be thinking about. And this would be, at least as of right now, we haven't used any maintenance strategies with these approaches. And that's something which patients value tremendously. So if we can get away with deep, durable responses without the use of any other drug, that would actually even justify the cost of these products upfront, because the cost of myeloma therapy, as you all know, and I've talked about quadruplets right now, each of those medications costs a tremendous amount, and a year's worth of myeloma therapy can make us bankrupt very, very quickly.
Thank you, Dr. Raje, for that interesting perspective on one round of therapy and nothing else after that. And we hope that at least some patients do receive that form of benefit from cell therapies in 2021 and beyond. And I think on that note, we just have 2 minutes left. So I'll just say my thanks. Thank you to all three of you. This was absolutely amazing. We covered a lot of ground actually in 1.5 hours. And to think about it, we went from cell therapies to COVID impact to bispecifics to AML, MDS, and all the exciting targets over there. So thank you. Wouldn't have been possible without your insightful inputs and your presentations. And we really look forward to engaging with you in the future. We look forward to doing many more of these kind of sessions in this new year and beyond. I'll hand it over to Hafiz if you have any inputs before we close.
That's great. Thank you so much, Dr. Raje. And we look forward to discussing again, and maybe at a later time, we can also talk about some of the COVID and pandemic related challenges that you're facing as well. So appreciate it. And then for the folks on the line, this will be available right away for you to preview at a later time as well. Thank you again.
Thank you. Thanks so much.
Thank you.
Thanks, everybody.
Thank you. Bye-bye.